Welcome 12th class students. This is Nanda biology tutorial. Today we will discuss that is female gametophyte. Now gynoecium is called to be the female part of a flowering plant, consists of stigma, style and ovary. We have discussed already. So ovary, inside the ovary there is a presence of ovule. Ovule is also called to be megasporangia. Ovule is also called to be megasporangia. So ovule, they are arranged on the structure that is called or cushion shape called placenta, and the arrangement of ovule is called placentation. We have discussed in 11. Now here the megasporangia or ovules, and what is the structure which further give rise to the female gametophyte. So in your syllabus you can draw, but in your syllabus the ovule is anatropous, means inverted ovule. For MCQ you can learn so many, but for the syllabus, inverted ovule which further give rise to female gametophyte. In your NCERT book this is the diagram, but inside we will fill that is the embryo sac structure also side by side. So this is inverted funicle, but here you will see this is called to be inverted ovule. So I have drawn inverted ovule or ovule structure. So the main structure, that means that this part, very important part, is called nucellus. Nucellus consists of parenchymatous cell, means living cell, which further go various division to form the embryo sac. So embryo sac is embedded in nucellus. Nucellus is protected by two covering, that is integument, outer and inner integument, which protect this nucellus. Outer integument and inner integument. Now this ovule remain attached to the structure called funicle. So funicle is the stalk of the ovule which attached with the ovary. Then there is a complete structure. This integument remain attached by means of the structure called hilum or helium of the chemistry. Hilum, H-I-L-U-M. Funicle is the stalk of the ovule, protected by two covering, outer and inner integuments. Integuments, that is the two covering which protect the nucellus part. Integument, this structure is complete but leaving a gap. Leaving a gap or pore is called micropyle. This micropyle you can observe. Suppose this is the gram seed, like this. So this black structure is called to be micropyle, which remain along with the next formation, female. So micropyle leaving the gap. Otherwise integument is complete, or protecting the nucellus, but leaving the gap is micropyle, a pore-like structure. Then the next point, this nucellus undergo the various changes and they act as further megaspore mother cell or MMC. Now this megaspore mother cell further undergo the division to form the structure. That means the mother megaspore form the structure. 4 are formed. 4 megaspore are formed. Out of these 4, 3 degenerate and 1 become functional, and 1 is functional, which further give rise to the embryo sac. So embryo sac is called to be the female gametophyte. It is called female gametophyte. Next, now this embryo sac or the further divide, this functional megaspore divide into mitotic division. First, there is a formation of the two nucleus. How these structures are developed inside two nucleus, and these two nucleus migrated to both the ends. It is given in your NCERT also, can come in the paper. That is how these embryo sac having the cells. So there is a development you have to learn. Diagram can come in the NCERT or in board. So then divide. Next division, 4 nucleus are formed. Next diagram, 4 nucleus are formed. This, then first is 2 on both sides, 4. Then next conversion, 4 on both sides, because they are dividing. So 4 on both sides. It means total 8 such structures or 8 nucleus are formed. What they form? Out of these 2 or out of these 8, 2 migrate in the center and rest form the structure on both the sides. What is it? That means 2 in the center called polar nuclei, and they form a central cell. Next point, rest of the 3 above, 3 below. This end is of the ovule called micropyle. Opposite end towards this micropyle is called chalaza or chalaza, your pronunciation, chalaza end. Now, these cells, they start migrating. And how embryo sac is formed? We will draw separate, 2 in the center. Then, if it is a micropylar end, or it is micropylar, where the micropylar you will draw, egg should be there towards micropylar end. If it is micropylar or micropyle side, egg always toward that side. And opposite to that form 3 antipodals. So it means egg cell, which is large, protected by 2 cells called synergids. Center cell is called polar nuclei, and 3 at opposite end, that is a chalaza end, is called antipodals. So it means in your paper or in your board, both the diagram can come. If it is come ovule, you will draw integument along with it. If it is come embryo sac only, then draw only this much part. If you will draw ovule along with, you will get 0. It means embryo sac a separate diagram and ovule is separate. This is a complete anatropos. In NCERT, only the nucellus is given, but the embryo sac structure is not properly given, but you have to draw for the paper. Very important. So, egg cell, large, synergid, polar nuclei, then 3 antipodals. It means 8 nucleus and 7 cell structure of embryo sac of angiosperm. Mostly it is a common structure, but variation is there, different. But we will learn anatropos means inverted ovule. If it is campy, means curved ovule. There are so many. But so now what is the structure? That is nucellus protected by 2 integument, outer, inner, leaving 1 gap called micropyle, and opposite end is called chalaza end. The development or inside the cell structure, 1 egg cell, 2 synergid, 2 polar nuclei, and 3 antipodals together constitute a embryo sac, or it is also called to be female gametophyte, which further take part in reproduction. We will discuss, that is a fertilization and pollination. So 7 cell and 8 nucleate embryo sac of angiosperms. Thank you.